breastfeeding and the role of early life microbiome in shaping the infant gut resistome. And the results that I will present today are part of a Nature Communications paper that I published during my PhD in the National Spanish Research Council. I am sure that all of you know that antibiotic resistance is the ability of bacteria to develop mechanisms to counteract the effects of an antibiotic. This natural evolutionary process of bacteria has been accelerated in the last decades due to the inadequate prescription, self-medication, overuse, and prolonged use of antibiotics. And the consequences of antimicrobial resistance is that the treatment with antibiotics is less effective, more difficulty on treating the infection, which leads to longer hospitalizations and a higher probability of sepsis. The World Health Organization has classified antibiotic resistance as a global public health threat and one of the top 3 challenges of the 21st century. Just in Europe, it is estimated that more than 30,000 deaths per year are caused by sepsis due to resistant microorganisms. And furthermore, in a paper published last year in the Lancet, this paper predicted by 2015 there would be over 40 million deaths per year caused by antibiotic resistance, surpassing those deaths from cancer or many other diseases like cholera too. And the most affected regions would be those less privileged, such as Asia, Latin America, and Africa. So this is why now is the moment and the time to act against antimicrobial resistances if you want to prevent this scenario from becoming a reality. Early life is a critical period for the acquisition of resistances. It is estimated that more than 200,000 infant deaths each year are linked to antibiotic resistances, especially those from low and medium income countries. And AMR is particularly concerning in newborns because their immune system is still developing, and infections can have both short and long-term consequences. It is also a crucial period for the acquisition of resistances since the infant gut microbiome is developing during that period, and the gut microbiome is considered the principal reservoir of antimicrobial resistant genes. So in early infancy, we can talk about the gut microbiome, but we can also talk about the gut resistome and the gut modulate. The gut resistome is defined as the collection of genes, their precursors, and mechanisms in the microbial community that confer resistance to antibiotics. And the modulome is the collection of all mobile genetic elements such as plasmids, transposons, bacteriophages, or integrons that facilitate the horizontal transfer of genes between bacteria. However, there is still a lack of knowledge of how the infant gut resistome is established and how the factors that the infants are exposed to might shape this resistome. And that's why the main objective of our work was to characterize the evolution of the infant gut resistome during the first year of life, study the role of the microbiota on its development, and understand the effect of perinatal factors on the acquisition of antibiotic resistance. For that, we worked with a total of 66 mother infant pairs from the MAMI cohort, from which we collected fecal samples and breast milk samples at 4 different time points, at 7 days, 1, 6, and 12 months postpartum. And we performed metagenomic sequencing on those samples. And we also considered the clinical data that we recorded, and also we considered the information regarding the infant delivery mode, their mode of feeding, and the period of weaning. So going deep into the results, the first main conclusion was that the infant resistome tends to resemble the maternal resistome. The diversity of the infant resistome increases during the first year of life till it resembles the mother's resistome, and the same pattern was observed regarding the resistome composition. But for the antimicrobial resistant gene load, we observed that it decreased during the first year of life. We observed that those infants at 7 days of age had a higher abundance of antimicrobial resistant genes than the same infant when he was 1 year of age. And even they had more antimicrobial resistances than their own mothers. As we perform metagenomic sequencing, we were able to track which were the microbial hosts of these resistances. And for example, we found that Streptococcus were the species that mainly carried tetracyclines and macrolide resistances. Beta-lactam resistances were mainly found and carried by Klebsiella and Staphylococcus, and aminoglycoside resistances were carried by Staphylococcus, Escherichia, and Enterococcus. But we were not only able to describe which were the microbial host of these antimicrobial resistant genes, but also if those genes were found in mobile genetic elements. And in our case, we specified if they were found in or associated with plasmids. And for example, we found that the resistant genes BLAZ and BLAZ10, which confer resistance to beta-lactamases, were mainly found in plasmids. For the case of BLAZ, it was mainly found in plasmids of Staphylococcus, and for the case of BLAZ10, it was mainly found in the plasmids of Escherichia. So with that, we know that those genes have a high potential to be horizontally transferred between bacteria in the infant gut. Regarding the establishment of the microbiome during the first year of life, and also based on our previous results, we applied K-means clustering and observed that the infants of our cohort clustered into two different groups regarding their microbial composition. And when studying the difference between these two groups, we observed that one group had a high abundance of Bifidobacterium, over 70% of relative abundance, and the other group, had a low abundance of Bifidobacterium, below 30% of relative abundance. And we observed that these two groups were consistent during the first year of life. And that's why we named one group as high Bifidobacterium and the other group low Bifidobacterium. And we could conclude that Bifidobacterium abundance determines the microbial composition during the first year of life. And going one step further, we also observed that those infants in the high Bifidobacterium group had less antimicrobial resistant genes than those infants in the low Bifidobacterium group. So in other words, infants with low Bifidobacterium abundance had higher abundances of antimicrobial resistant genes. And also those genes were more associated with mobile genetic elements such as plasmids. So not only Bifidobacterium abundance determines the microbial composition of the infant gut during the first year of life, but also determines and is associated with the resistome load and the modulate of the infant gut. And as I told you, one of the main objectives of this work was also to study the effects of the different factors to which the infant is exposed that could shape the infant gut resistome. And we hypothesized that the same factors that shape the gut microbiome of the infants could shape the infant gut resistome. And we observed that in early time points at 7 days and 1 month of age, factors such as the mode of delivery of the infant and the diet that they were receiving, if they were receiving exclusive breastfeeding or not, were the factors with the strongest effect on the acquisition and the composition of the resistome. So, focusing on these two early time points and these two factors, we observed that at 7 days of age, mode of delivery was the factor with the strongest effect, and we observed that those infants born by C-section had a higher abundance of antimicrobial resistant genes than those infants born vaginally. And also, the resistome composition was different regarding mode of delivery. But at 1 month of age, mode of lactation was the factor with the strongest effect, and we observed that those infants that were receiving exclusive breastfeeding during this first month of life had less antimicrobial resistances than those infants that didn't receive exclusive breastfeeding, because they were receiving formula feeding or a combination of formula feeding and also breastfeeding. And in our cohort, we had all 4 possible combinations of this. So we had infants born vaginally that received exclusive breastfeeding, but also that didn't. And we had the same combination for the C-section born infants. So we wanted to study which was the effect of these 4 combinations on the resistome load of the infants in our cohort. And we observed that at 1 month of age, as expected, those infants born vaginally, but that received exclusive breastfeeding, had less antimicrobial resistant genes than those infants also born vaginally, but that didn't receive exclusive breastfeeding. So we observed the effect of exclusive breastfeeding. But interestingly, we observed that those infants born by C-section, but that received exclusive breastfeeding during the first month of life, had less antimicrobial resistances than those born vaginally, but that didn't receive exclusive breastfeeding. So with this, we could conclude and observe for the first time the potential role of breastfeeding in reducing the effects of a C-section delivery on the infant gut resistome. So with these results, we could conclude that the dynamics of the infant resistome during the first year of life tend to resemble the maternal resistome. We observed that the high abundance of bifidobacteria is associated with a lower resistance burden. We observed that delivery mode and breastfeeding are the two factors with the strongest effect on shaping the resistome in early life. And we also observed that exclusive breastfeeding may be a strategy to reduce the effects of C-section delivery on the infant resistome. And with these conclusions, future research should focus on high relevant clinical antimicrobial resistant pathogens that affect newborns in different hospital settings. Also, future research should focus on those infants that are more vulnerable, with lower access to antibiotics but highly exposed to resistances acquisition, such as newborns from low and middle income countries and also preterm infants. And then next research should focus on deciphering the molecular mechanisms behind all these associations found when studying these results from big cohorts to ultimately develop translational strategies to combat antimicrobial resistances from early stages of life. And just to finish, I would like to thank my PhD supervisor, Maria Carmen Corleado, who supervised this work and led the MAMI cohort. And thanks to all the audience for your attention. Thank you very much.